Okay, let's have a class. And today's class, we're going to do aseptic transferring bacteria, and we'll start to work on the unknown number one. That will be the homework, and you bench partner work together. So we're going to have a lab 4, lab 5, lab 5. So we'll say bacterial transfer. And unknown number one. So before we talk about bacterial transfer, there is one concept we wanted to introduce to you called the aseptic technique. What means aseptic technique? This means transferring wanted microorganism into a target medium without carrying contaminants. So this is very important. If we are transferring a bacteria on a slant in the broth or onto the agar plates, if you're transferring E. coli, then your transferring is only E. coli. You're not going to carry on the bacteria from the air on the bench from your hands and go to this medium, because otherwise you're going to get cross-contamination. So that's the aseptic technique, the concept. Okay, the second thing, we want to introduce you what type of the medium we will have, and we mentioned in the lecture, but here we're going to be talking about these a little detail. Okay, we have a liquid medium. The liquid medium is broth. It looks like this. Little bit of light yellow color, looks like a tea. It's the same color. Okay, what does the broth looks like? The broth is a liquid medium. So the ingredients, obviously, you should have salt, sodium chloride. They will have beef extract because they give them a very high nutrition. They definitely have sugar. And usually the sugar, either glucose or dextrose. And they also have a combined some thing from a protein. We call it a peptone or tryptone. And very important, of course, there is the water. Okay, so the major ingredients looks like give them a very high nutrition for bacteria to grow. Now we have to emphasize that the bacteria grow in the broth. Usually we call it vegetative cells. That's how normal bacteria are going to grow. But the broth, we also call it a nutrient broth. They cannot grow fastidious bacteria. Let's say Campylobacter jejuni. They won't grow just you put them into nutrient broth. They need a very strict environment, also a special medium. So that's something I want to tell you. This is a vegetative cells, not fastidious bacteria. Okay, so this is the first one, it's a broth. The third thing we want to mention is the agar. Okay, agar is a solid medium. And this is from the polysaccharides of seaweeds. This is we already talked about. And we mentioned in our class, in the lecture, this is Miss Fanny Hesse, and the first person who is find that the polysaccharides from the seaweeds is making an agar is a good material, because these kind of the material will be melted at 50 to 55 degrees Celsius and solidified at room temperature. And this is very important. Now basically there is a two type of the agar we're going to use in the lab. One of them is on a tube, and this is called slant. And slant, what we do is temporary storage bacteria. This is usually at 4 degrees Celsius, maybe about like 7 to 10 days. Just temporary period. So when you have a slant, we're going to do a zigzag streak. Okay, that's using zigzag streak. Now another one you will find is on the Petri dish. This is Dr. Richard Petri created the container. It's called agar plates. And this agar plates, what we can get is pure culture and slow streak plating. Now obviously later on you will know the agar plates could do pour plating, spread plating, and will get single colony, could do numeration. This we will do next week. Okay? So this is some of the introduction. Now today's work, we're going to move on to today's work, is very straightforward. However, this technique you have to know. Okay, number one is for broth. How to do? You will get a Bacillus subtilis slants from last week. Pick the loop and then put into the broth. But there is one thing we have to be emphasized. Start from today, you are doing lots of the tubes and the plates. What is the important thing you do? Is label. You're going to add a 3-star there. Label is very important. If you do not do a good job of label, we have nearly 40 students in this lab. You're going to spend half an hour to find your plates and your tubes. So the label is very important. If we have a broth, where you should label? Let's say this is a broth, okay? Your label is always on the top. Label your initial, date, and the bacterial acronym. EC is EC, BS is BS, for example. And you could put your bench number. These are the information you should put. Okay, that's the first thing. So you're going to pick the bacteria from the slants and then go in here. And the tube will be storage 35 degrees Celsius, 24 hours. This is the first thing to do. Very simple. Second thing is a slant. Okay, slant is also very simple. You have a slant like this. This is a slant. And where you should label? Right on the corner here. Not on here. If you label here, you're going to cover the whole thing of the slants. You cannot see whether it grow or not. So right on the corner here. So the slants, if it looks like this, you're going to label right here. So what you're going to label? Date, bench number, your initial, and the bacteria's name on this corner. And we also going to pick from here. We'll do a zigzag streak. So that's called a zigzag streak. This is also, we're going to put 35 degrees Celsius, 24 hours. We hope to see either turbidity, precipitation, or pellicles for broth. We'll see a very clear zigzag marker for slant. Okay, now the third one is agar plate. Okay, this is something is important today. Today you will get a mixed culture. They basically have two bacteria there. One is E. coli, another one is Serratia. You will see very interesting. They are showing you two different colors after you grow. So what we will do, basically we have 2 days, we'll just put this at room temperature. Because Serratia at 25 degrees Celsius is supposed to be a very pink red color. And the E. coli is just a normal colony, colorless. So I think we'll just put in the room temperature for 48 hours. You will see the culture will be showing up. All what we can do is that we certainly could do is that we put 35 degrees Celsius for a day. Another culture we're going to be put in the room temperature. So hope to we'll see the difference. Okay. So this is how we're going to do. It's the agar plates. And a very simple question, where we should label. There is lid and there is agar plates. Where we should label. You should always remember, you have to label at the bottom of the agar plates and never on the lid. Okay, so the label is at the back of agar plates. Never on lid. Why? Because for somehow this agar is on the floor. You're going to be separate then. If you label on the lid, you never know which one is this. So always remember, I have to label at the bottom of the agar. Or sometimes you're working the other labs, they may ask you to label on the edge. That is fine, but definitely not on the lid. Okay, so that's the first thing. Now, what are the information we're going to do? Same thing, the information is the date, initial, the bacteria name, you can just say unknown today. And very important is your bench number. Okay, that's number one. Now, what we're going to do? You will get is this is the unknown. Okay, this is the unknown is a plus. We're gonna have to do is called streak plating. So what is that? You will pick some bacteria use loop right here. Okay, then in here, very, very gentle to do a first conj. Then flame you loop. At this edge, do a second less intensive streak. Then flame you loop again. On the edge, to do a third less intensive streak. Then flame you loop again to do a last one. Every corner have two flaming loop. Every corner. Every corner. I'll just say every corner. Okay. And this is very important. What you have to flame the loop, because this plates we will be incubating 35 degrees Celsius 24 hours. Ideally, you should see a single colony like here, like this, like that. And ideally, you should also do two different colonies on the agar plates, a different color. This is a single colony. Represents a genus name, a species name, this whole thing represents a pure culture. But you keep in mind, a single colony does not mean one single cell. It's a colony. So how many bacteria are there? Roughly at least more than 10 to the power of 6 to 7. Okay? That's keep in mind. Does not mean it's a single cell. It's a cluster. Okay? Now, streak plating is very important because this is what we always say. It's like serial dilution on solid agar medium. So what we do? Two of you maybe need to share. Okay, a plates, a slants, a broth. Make sure it is shake. Shake first, mixed. Then you do the streak plating. Then you hopefully, after incubation on Thursday, we want to see two different colonies. So this place is important. From the two different colonies on Thursday, we will do two things. Number one is description of colony morphology, the colony morphology will be the slides on your lecture. So I will be mentioning this. This is lecture slides. I will try to print out for you, but won't be color. Second is gram stamp. Differentiate two colonies. Now, sometimes you may get only one colony. Or on your plates, the colony the same. That's fine. Sometimes we get something like that, then you can describe that. And then finish, complete. Is two people per group report. Okay, this we will talk about on Thursday, this section. Okay, so this is what we have, same as before. We'll be showing you how to do this. So I will pick plates, slants, agar, and the mixed culture. We're gonna move to here, the same thing. We'll be showing you how to do it. This is the first time we're using the real agar media. So I hope everybody will get it. It is very important. Okay? This is the first time you touch real bacterial media. Although we did a couple of the staining technology already. So we want to emphasize something. First thing first, these agar plates and the broth are just coming out from the refrigerator. What this means? Humidify could be. So what you do? Dry it. Otherwise, your marker cannot put on. Okay, all these you should be dried. Okay? You see that? Agar plates, very important, dry. If you see liquid on the edge, clean it. Okay? Because it's going to bother you. The condensation will bother the results. That's the first thing first. Second thing is label. It's very important. Where are we going to label the broth? On the top. So initial batch number, it's going to be Bacillus subtilis and date 0, 9, 10. At least this information. Where we should do the slants is right on the edge, not here. You're not going to see it. Okay, so on the edge you're going to do initial batch number. So that's your initial, it's not my initial. So you're not going to everybody write CS. Your initial is not CS. Then that means nothing, okay? Make sure your initial. Then batch number is not number one. I just give you an example. What is your bench number based on you see on your bench? Okay, now in the bacteria name, the Bacillus subtilis and date 0, 9, 10. So what you're going to look on Thursday, you're going to look is clearly is on the top. Now the agar plates, we mentioned never label on the lid, so you have to label at the bottom. So we're going to label is this my initial bench number. This will be unknown and also will be 0, 9, 10. Okay, so these are the things we're going to have to do the label before we do. Then we're going to transfer the bacteria. Obviously, we need the Bunsen burner. So make sure the Bunsen burner, it works. So we're going to try to turn on the Bunsen burner. Okay, that looks good. And then make sure your loop is good. This is very important, is streak and really have a loop on the top, okay? Sometimes you will see something like this. This is called a needle. Will be used on the later lab for sulfide indo motility test, but not today. So make sure you have a loop on the top. Okay, now the first thing is sterilization. Make sure the whole thing is red and a little bit on the here. Okay, now we're going to transfer from slants to broth first. Little finger turn on, cool down, pick some bacteria, flame it, put on the side, and in broth it's very simple. Little finger, flame, then go in, shake it, flame, that's it. And we will be incubate 35 degrees Celsius. We'll see what we get. Okay, now flame the loop again. Little bit here. Then what we do? Go to the slants. Okay, Bacillus again. Hold on. Pick some bacteria on the side. Flame it. Go here. What we do is zigzag streak. So flame the loop. Don't put the button, the bottom. Okay, just right here. Very gentle zigzag. Don't block the agar. Flame down. That's it. Okay, now this one is your homework, so it is important. Flame your loop again. First, make sure Bacillus is gone. Now, this one, make sure you shake it. Make sure you shake it very well. I will shake 15 seconds. Okay, because a lot of them at the bottom. So flame your loop again. Okay. Now, hold on. Go to the broth. Then, we're going to do streak plating. Okay. Very gentle at the beginning. Flame the loop. Hear the voice. Then on the side, second streak. Flame the loop. Hear the voice. Then go turn. Right here. Third streak. Sometimes I do third in the lab, but you can do third or fourth. Third quadrant, fourth quadrant, that's both are fine. Then hear the voice on the side, do a last one. And you can see you have a marker there. Okay, you have a 4 quadrant marker there, then that's good. Okay, now make sure, turn it off. I will keep my plates, because I know some people won't do very good. Then I can borrow my plates to them. Okay, so I keep my plates here. And also, when you finish everything, make sure in a good order, give to TA. Otherwise, it's going to be a completely mess on Thursday. I think he will arrange a rack for blocks, a rack for slants, another special one for agar plates. It is simple, but you have to do in a civil way. What I mean a civil way is that organized, gentle and organized. Because later on, especially when we move on to biochemistry test, you may have 5 tubes in one lab or 7 tubes in one lab. If you do not do a good job of label, not organized, you're going to spend 20 minutes to find your plates or the tubes in the next lab. That's happened all the time. One more thing. This is called king cap tube. It's very easy to fall off, so never hold on like this. Make sure you have 2 hands. It's very easy to fall off. This is a number 2 accident in the biological lab. Number 1 is a Bunsen burner or diode, those things. Very easy to fall off. If you fall off, let us know. We ideally should be put in a Sharpie glass box. You cannot just put it into the biohazard trash can. Okay? Something important. Okay, now go ahead to work.